A world-first breakthrough, using a woman's own stem cells to reverse type 1 diabetes. In a groundbreaking medical achievement, a woman's own stem cells have been used to successfully reverse type 1 diabetes T1D, a condition previously considered irreversible. This remarkable breakthrough represents a major step forward in regenerative medicine and could pave the way for a potential cure for millions of people suffering from the disease. Below is a detailed explanation of this breakthrough. 1. Understanding type 1 diabetes. What is type 1 diabetes? Type 1 diabetes is an autoimmune disease in which the body's immune system mistakenly attacks the beta cells in the pancreas. These beta cells are responsible for producing insulin, the hormone that regulates blood sugar glucose. Without insulin, blood sugar levels rise to dangerous levels, leading to severe complications such as kidney failure and nerve damage. Graft diseases, increased risk of infections. People with T1D are required to take lifelong insulin injections or use insulin pumps to manage their blood sugar levels. Why is type 1 diabetes so hard to cure? Unlike type 2 diabetes, which can sometimes be managed with lifestyle changes, T1D is caused by the complete destruction of beta cells. Scientists have tried various approaches, such as pancreatic transplants, stem cell therapies, and immune system modulation, but none have provided a permanent cure. 2. The stem cell therapy breakthrough, were stem cells used to reverse type 1 diabetes. In this historic medical breakthrough, doctors extracted stem cells from the woman's own body and reprogrammed them into insulin-producing beta cells. These lab-grown beta cells were then transplanted back into her pancreas, where they began producing insulin naturally. Why is this breakthrough significant? First time a patient's own cells were used. Previous studies used donor cells or lab-engineered cells from other sources. This is the first time a patient's own stem cells successfully reversed T1D. No need for immunosuppressive drugs. Since the cells came from the patient's body, her immune system did not reject them, eliminating the need for lifelong immunosuppressants which come with significant side effects. Restoration of natural insulin production. The patient's body started regulating blood sugar levels naturally without the need for external insulin. Technical details on using stem cells to reverse type 1 diabetes. This groundbreaking case represents a significant advancement in regenerative medicine, cellular reprogramming, and immunology. Below, we break down the intricate scientific processes involved in this breakthrough. Type 1 diabetes and beta cells. Role of beta cells and their destruction in type 1 diabetes. Beta cells, located in the islets of Langerhans within the pancreas, are responsible for insulin secretion. In type 1 diabetes T1D, the immune system mistakenly identifies beta cells as foreign invaders and destroys them via autoreactive T cells CD4+ and CD8 lymphocytes. This results in absolute insulin deficiency, leading to hyperglycemia and metabolic dysregulation. Current treatments, such as exogenous insulin administration, do not mimic the precise glucose regulation of natural beta cells. 2. The stem cell therapy approach. Step 1. Isolation of the patient's autologous stem cells. In this case, autologous stem cells derived from the patient's own body were used, avoiding immune rejection. Two potential sources of stem cells were considered: hematopoietic stem cells HSCs from bone marrow or peripheral blood, mesenchymal stem cells MSCs from adipose tissue or bone marrow. These stem cells were then isolated and expanded in vitro. Step 2. Differentiation into insulin-producing beta cells. Isolated stem cells were subjected to a multi-stage differentiation protocol to transform them into functional beta-like cells. The differentiation process was influenced by specific transcription factors, mimicking pancreatic embryogenesis. Endoderm induction cells were treated with activin A and WNT3A, inducing differentiation into definitive endoderm. Pancreatic progenitor stage. The addition of FGF7 fibroblast growth factor 7 and retinoic acid Ra promoted the transition to pancreatic progenitors. Beta cell maturation cells were exposed to ING3, PDX1, and MAFA, key transcription factors responsible for insulin production and glucose responsiveness. The final stage involved treatment with GLP-1 agonists and glucose to enhance insulin secretion functionality. Step 3. Cell encapsulation and protection. To prevent the immune system from attacking the new beta cells, they were encapsulated in a biocompatible hydrogel scaffold. This encapsulation technology allows for nutrient and oxygen exchange while protecting the cells from immune attack. Eliminates the need for immunosuppressive drugs, which are typically required in organ transplants. Step 4. Transplantation into the pancreas. Beta-like cells were injected into the pancreas or implanted in subcutaneous tissue if pancreatic access was not feasible. The transplanted cells successfully engrafted and integrated with the patient's existing pancreatic islets. Step 5. Functional testing and monitoring post-transplantation. The patient was monitored using C-peptide levels, a marker of natural insulin production, glucose tolerance tests (GTTs) to confirm normal insulin response, continuous glucose monitoring (CGM) to assess real-time glucose levels. The patient exhibited restored glucose homeostasis, confirming functional insulin secretion. 3. Challenges and future directions. Framing challenge: long-term survival of beta cells. Although encapsulation helps, long-term survival of transplanted beta cells remains a concern. Autoimmune response could still target new beta cells if the underlying immune dysregulation is not addressed. Scalability and standardization. Transplanting patient-specific stem cell-derived beta cells is time-intensive and costly. Mass production methods, such as induced pluripotent stem cells (iPSCs), are being explored for large-scale application. Immune system modulation to ensure a permanent cure. Addressing autoimmune attack is crucial. Strategies like regulatory T-cell Treg therapy or tolerogenic dendritic cells may help prevent further immune destruction. Future implications: if refined, this therapy could eliminate lifelong insulin dependence. Gene editing technologies. CRISPR-Cas9 could be used to create genetically modified beta cells resistant to immune attack. Bioartificial pancreas technologies combining stem cells and encapsulation devices may offer a permanent diabetes cure. 4. Conclusion: this revolutionary autologous stem cell therapy is the first successful case of reversing type 1 diabetes using a patient's own cells. While challenges remain, this breakthrough provides a foundation for future regenerative treatments that could eventually cure diabetes at a large scale.